So it's this whole process. And I always say the soil is the gut, and the microbes control the uptake and flow of nutrients. And I could buy a NASCAR to buy groceries, to go get groceries, but it's probably overkill.
But for decades, the way we've tested our soil hasn't really changed. We've treated our fields like a sterile chemistry set, but as any grower who has stepped into the world of regenerative agriculture knows, soil isn't a factory, it's a living, breathing ecosystem. The big question has always been, if the soil is a living engine, how do we look under the hood and measure its performance? We're gonna hop on today with Lance Gunderson. He is the co-founder and president of Regen Ag Labs. Lance, it's an honor to have you here today. Welcome to the show.
Oh, thank you so much, Brandon. Yeah, looking forward to this discussion today. It's going to be pretty fun.
Absolutely. You know, why do we spend so much time, and this has always been my question, right? And everybody's question, I think. We spend so much time looking at the plants, but so little time looking at the engine. And right, that's the soil, what's driving them. And, you know, for the longest time, we always look at plant health. Oh, how are the crops doing? But a lot of times we forget to actually look at the soil and what's going on there.
Look, I get asked all the time, to either define regenerative agriculture, or people are commenting about how there is no definition and it changes. Well, the way I look at it is like this, is that the definition does change. But if I had to wrap it into something, what I always say is this, is that, look, regenerative means you're bettering the soil tomorrow than where it is today. Now, how you define better, again, that's a little up to you, right? But when you get up in the morning and you look at yourself in the mirror and you say, I'm going to try to be a little better today than I was yesterday. Whether that's you're going to exercise or eat a little different or take time to spend more time with your kid, whatever it is, however you're going to define that. But really, in a nutshell, it's just about taking a step forward for your farm, and how that needs to happen and how we measure it then is a little dependent upon your definition as a grower or as a company, as a consultant. And that's the approach we try to take.
Well, but with inputs that you're putting in to make that system better, you really have to think twice. Is it going to make me better next year? Is it an investment into my farm, or is it just a direct expense that I really have no, extra benefit as years to come?
Sure. Well, and even with the testing itself, they can, soil tests can be just another cost that gets you nothing. Because if you're not aligning the test with your goals, it doesn't do you any good. I'm an analogy person. You're going to hear a lot of them today. But, if you wake up in the morning and your shoulder hurts and it's been hurting for months, and you go to the doctor and they take an x-ray of your knee, well, they're running tests, but they're missing the whole point, right? And so you're wasting your money, you're wasting your time. And the reason we really try to get people to define regenerative agriculture for themselves and lay out their goals is because we really want to try to provide tests that are going to impact those goals. It's not just to collect data. I could do that all day long. But at that point, it's just another input cost to you as a grower. And
why the standard salt extraction, traditional soil test, right? For years, that was the only thing was, ever ran. And it often really fails to show the whole picture of nutrient availability, right? So if you could, can you just kind of explain the problem with the traditional soil sample?
Well, before I say this, let me point out, those tests were helpful. They got us somewhere. In the world of agriculture. If you go clear back into the 40s and 50s, after years of nutrient removal, growing crops, fertilizers, soil testing to determine fertilizer application, it helped. It helped agriculture. It increased production. There was a lot of good that came out of that. We can say the same thing about the original telephone. But times change, technology changes, the science and understanding changes, but the testing has stayed the same, right? And so it's not that those tests are not useful in certain situations or anything like that, but they have limitations. And primarily, those tests were following the idea that your soil operates as a chemistry set. So when we talk about chemistry, if you've ever taken chemistry classes, it's all about controlling the ratios of different things in a beaker, the temperature, and all of these things in order to get this reaction to happen. Well, the reaction we want to happen in agriculture is that we're going to add all of these inputs into the beaker, and the soil is the beaker, and then we want this reaction to happen where the plants take up these nutrients and produce this crop. Well, you're dealing with a biological system. And so when we just focus on nitrate and phosphate and ionic potassium and calcium, etc., we're kind of missing the idea. We're kind of missing the idea. Now, the analogy here is the human gut. You know, for years we thought, well, if you just eat nutritious food, if you take vitamins, you know, your small intestine absorbs all of that stuff and makes you healthy. That's how. Now your small intestine there is the plant roots. You know, we put all this stuff on, the plant roots just suck all this stuff up, and we've got this healthy plant. Well, that's not really how it works, right? Your small intestine is where the microbes live, and the microbes control the uptake and flow of nutrients. And yes, they need to be fed good things. That's why fiber is so pushed in diets anymore, right? Fiber, because really you don't need fiber. You're feeding the microorganisms. And so it's this whole process. And I always say the soil is the gut of the system. And so, yes, what we put into the gut is going to dictate what comes out. So the issue with some of these tests is that we're only really measuring nutrition like looking at a multivitamin. Fertilizers are multivitamins. We're going to add the N, the P, the K, the iron, the whatever it is. Well, that doesn't really mean you're going to get good absorption, right? You're not going to translocate that into the plant. We got to focus more on the microbes because they're the ones that are doing this work. Now, with that being said, do we still need to make sure that there is phosphorus in the system? Sulfur? Absolutely. You know, and the big difference in an ag system versus a native system is that we have huge amounts of nutrient removal.
Absolutely. And then that's kind of what's now brought us to, you know, I guess the past 15 years kind of what you've been pushing out, really focused on the Haney test, correct?
But the big difference is that the Haney test, I would say it's a three-leg stool. On one leg, we're going to evaluate soil fertility. The biggest difference there is that the extract being used is what's called H3A. Dr. Haney came up with that, developed it over a span of several years, many different iterations of it. But at the end, his goal was to create an extract that A, doesn't overpower the soil pH. So, you know, we all know the limitations of Malic 3, especially out in the western states, high calcium soils, calcareous, high pH. The idea was not to overpower the natural environment. Number two, it was to use ingredients, if you want to call them that are naturally found in soil solutions. So there are 3 organic acids used in that extraction. They are organic acids that plants produce and exude out through the roots. We're using that to try to mimic soil solution. Now, yes, there are hundreds of different compounds that plants produce.
Sure.
Through a literature review, he picked these 3 because they were more common. So that's one leg of that stool. So that's how we're evaluating fertility. Everything else in the lab is the same. The same instrumentation, same color development processes as Malic 3 or Bray or anything. I mean, it's the same. Then we've got a water extract to look at how much carbon is available in water, soluble in water. So this is a subset of organic matter. We're looking at soluble protein, organisms that eat, so everything outside of plants and things that can photosynthesize and a few microbes, everything that eats basically consumes protein. Nitrate is toxic at some level to all of us. We eat protein, and much like ruminant animals, the very first thing they digest is soluble protein. Well, who's doing the digestion? The microbes. So we know how important soluble protein is to them, and we're able to look at nitrogen differently that way. And then the third piece of the puzzle is soil respiration, where we're looking at how much your soil essentially breathes, or what is the demand of carbon through the microorganisms. Microbes produce CO2, living organisms do through respiration, and that's what we're looking at there.
You know, why it matters for carbon sequestration and nutrient cycling, that fungal bacteria ratio.
Yeah, so the fungi to bacteria ratio has kind of been a popular topic in soil health since the dawn of soil health. The biggest thing is that fungi and bacteria behave very differently by and large. So bacteria are a little bit like, when it comes to carbon, bacteria are like sprinters and fungi are more like marathon runners. So fungi have a tendency they do so many things, but aggregate soil, but store carbon. They're kind of like perennial plants, if you want to think of it that way, versus annual plants, which is kind of like bacteria. They're just acquire as many resources as quickly as we possibly can, reproduce as fast as we can, right? And perennials are a little more long haul. So the ratio, you hear, if you go out, you hear all these different ratios, and they say, 1-to-1. Well, 1-to-1 measured how? Is that like a pound of fungi versus a pound of bacteria? Like, if you could get those out of the soil, is it a pound of carbon in bacteria versus a pound of carbon in fungi? Because depending their carbon content. So
yeah,
the ratio is great. I'm only throwing that out there because we need to understand the context of what they're talking about when they say ratio. The PLFA test, for example, measures a fungal to bacterial ratio. I know we'll talk about DNA in a little bit. DNA can do that as well. Certain tests can, but the ratio, the context of the ratio is different. So, for example, PLFA. If I had a pound of bacteria sitting here and a pound of fungi, and I extracted those and measured PLFA, bacteria, pound for pound, have more PLFA in them than fungi do. So even if I have a pound of each and I measure the ratio using PLFA, I'm not going to get a 1-to-1 relationship, right? So if you've ever seen one of our PLFA tests, you'll notice that when you get to about 0.25, which is 1-to-4, so fungi 1, bacteria 4, or 0.25, that is where we're starting to get a more equally established balance between the two. We get above that, and we're really getting close to that 1-to-1, and we can even get a little higher than that. Again, if you were going to measure it with the other ways. There are limitations. When it comes to management, of course, fungi do not like tillage. I'm not telling everybody here to not ever till, but those are some of the repercussions of those decisions. Fungi don't like tillage. No microbe likes fallow. Periods, bare soil between growing crops. Fungi especially. And mycorrhizal fungi are obligate symbionts, meaning they don't have a choice. They can only acquire energy through the association with plant roots, living plant roots. So they really don't like fallow. Got a pH up in that way high 7s, 8s. Bacteria have a slight advantage over fungi.
Absolutely.
It doesn't mean fungi won't do well, but they do have a slight advantage. On the other end of the spectrum, we get down in that 5.5, below 6, especially down to 5.5. Fungi tend to have a little bit of an advantage. And then there's obviously nutrient problems at both of those extreme pHs, right? But yeah, fungi, we used to think mycorrhizal fungi transported phosphorus. And that was it. That's what we always talked about, mycorrhizal fungi and phosphorus. Mycorrhizal fungi, though, they transport phosphorus, zinc, they propagate water, they move water in and create drought resilience. They actually form those Hartig nets and things around the roots to help protect them from disease. Think about it. That's their food source, right? The number one thing you can do to detriment your fungal population is actually fallow. I used to say tillage, right? You can never till. No, it's fallow. They go dormant. The problem is next year when you've got a crop, you're growing an annual, most of us, and I understand turf and things, but you've got an annual crop and you've got a 3-month window, 4-month window, 5-month, where you're trying to, again, what's the crop's goal? Acquire as many nutrients and resources as quickly as possible to produce the most amount of seed. Ironically, the plant's goal is the same as our goal. But if it relies on the microbial community to help do that, there's a lag. Yep. There's a lag period. So you want the seed to take off and germ and just run and go. Well, it takes a few weeks for the mycorrhizal fungi to recolonize and start. So by reducing the fallow, the power of cover crops is, you'll hear the term biological priming effect. You're bridging the gap.
Absolutely.
In other words, you're giving the crop a head start. Protozoans rely primarily on 3 things. One of those is food source. So you've got to have enough bacterial biomass out there to support them. Much like a lion on the plains of Africa, you got to have 10,000 wildebeest to support 50 lions or whatever, right? So they're always going to greatly outnumber, the prey is going to greatly outnumber the predator. Number 2, protozoans are truly subaquatic, meaning that they swim in water films around soil particles. So you have to have water and you need to have pore space. So good aggregation. And aggregation influences water as well. And then, number 3 is temperature. So as soils dry out, protozoans tend to form cysts and go dormant. The reason why they're so important, though, in the system, it has to do with carbon to nitrogen ratios. Bacteria, on average, have a carbon to nitrogen ratio somewhere between 3:1 and 5:1. What that means is that we think corn or wheat requires a lot of nitrogen, right? Corn at maturity is around 80:1. So bacteria are just little bags of fertilizer. They're very high protein, very high nitrogen. Protozoans have a C to N ratio of around 15:1. What that means is that as they graze and consume bacteria, they're ingesting 5 times more nitrogen than they need. Well, when that happens, they do exactly what every organism does that eats. They excrete waste products. And those waste products are nitrogen-based, ammonium, nitrate, that gets converted to nitrate, of course. So what we call that entire process is essentially nitrogen mineralization. And it is the back end of the nitrogen cycle. Plants take up nitrate and ammonium primarily. They build amino acids and amino sugars and proteins, etc., etc. Everything else eats them. And then convert out nitrate and ammonium again. And so having that system running is a big part of nitrogen efficiency. And much like plants can't just take up 200 pounds of nitrogen and hold on to it, right? And then move it around. They need a trickle charge effect. And so that is what naturally happens, is that protozoans are grazing and kicking it out slowly. Plants are taking it up as they grow, and it's a constant, like, two conveyor belts running next to each other. The problem is that in ag production, we expect that conveyor belt to run very quickly because we have a relatively short window, and we're trying to make a soil produce 300 bushel corn when it wasn't even designed to grow corn. But that's where the fertilizer supplementation comes in.
Sure.
But we've ignored that other conveyor belt for so long, and we've tried to just force feed everything into the plant. And again, we've increased production, but it's not very efficient.
No, not at all.
And then when you have poor efficiency, it's costing somebody money. And who's paying for it? The only people who win in that scenario are the people you borrow the money from and the people you write the check to. PLFA is going to measure biomass and what we call functional group diversity. We're able to break the organisms into large functional groups and look at that. But the limitation on PLFA is you can't look for individual species. The advantage is that it includes everybody because it's a community analysis where you're analyzing PLFA as a marker, as a molecular marker. DNA testing, now I will say this, not all DNA testing is created equal, and that doesn't mean useful or not useful. What I mean is there are so many different techniques out there, and I'm certainly not an expert in all of them or really any one of them, but there are companies that are using, like Biomakers, for example, uses PCR, 16S PCR. So they're able to isolate certain genes, amplify them, and then sequence them. And what they do with that sequence data is it's called blasting. So you blast all this unknown sequence data against a database of known sequences. And then you get a return and say, yep, this one matched this one matched that one, et cetera, et cetera. Using that technique, you're able to identify species as long as they're in the database. So as long as they're known, you can do that. Beyond that, there are markers that say, we don't know what species this is, but we know it's fungal or we know it's bacterial. So that's more like PLFA in that regard.
It gave you that functional group versus this is what the actual species is.
Yep.
Gotcha.
And then there are other techniques out there, what they call like whole genome shotgun approach. In a lot of those scenarios, they're not actually looking for the species. What they're doing is sequencing whole genomes, and they're looking for specific genes that provide a specific function. Similarly, we have to know which genes produce which function. So that has to be in a database, right? So we sequence a genome and we find all this series of A, T, G, C's, and U's, and we get this thing. Oh, and it matches this gene that codes phosphodiesterase, for example. Now, we don't know exactly which organism that gene came from. And in this case, we don't care. We care about the function. And so we're like, hey, we've got a whole bunch of phosphodiesterase. It's a strong marker. We quantify it. So your soil probably has the ability to cycle phosphorus better than a different soil that has much lower levels of that.
Sure.
So I always get asked, well, which one should I use? Well, that depends on your goal, right? I can buy a NASCAR to buy groceries and go get groceries, but it's probably overkill. The three tests that everybody kind of comes back and says, I really enjoy these is number one, it's total nutrient digest. So really quickly, what that test is that it is essentially a measure of your net worth when it comes to nutrients.
Sure.
We are going to categorize or quantify the amount of nitrogen, potassium, calcium, etc. that's in your soil. Most of that is unavailable. So much like sitting back saying, you know what, I looked at all my assets and all my debts and I've got says I'm worth $12 million. Well, congratulations. How much of that can you use today?
Yeah.
Very little maybe. So that's the idea of it, is we want to show you that your soil has more nutrients in it than what extract tests show you. Number two, we run the Haney test. And we say, okay, now we're evaluating what is actually available. So this is like looking at your checking account, right? So we're going to look at all these things. We're going to look at nitrogen, and we're going to look at nitrate, which is standard soil test. That's as far as it usually goes. Ammonium, maybe if you ask for it, but not many standard soil tests come with just ammonium. If you get east of the Mississippi, most standard soil tests don't even come with nitrate. They don't measure nitrogen at all. And then we're going to look at organic nitrogen or soluble protein. We're going to look at that. We're going to look at respiration and the balance between the carbon availability and the nitrogen. The organic nitrogen is part of that. I mentioned before that protozoans consume bacteria and then mineralize nitrogen. Well, if you're feeding a microbe something that's like a rice cake, all carbon, very low protein, they have a tendency to tie up more nitrogen in the system. That includes the nitrogen you apply as fertilizer. If your ratio is too low, they've got all this protein out there, but they can't mineralize it very fast because they don't have enough energy to do the work, right? So I always say salary and benefits. If you're looking, if you think of microbes as employees of your farm, salary is the carbon, the benefits is the protein and the right nutrition out there. You can't just offer one without the other, usually. Thirdly, then, is PLFA. Now, PLFA and TND, the Total Nutrient Digest test, those are generally baseline tests, year 0 or year 1. And then typically repeated every 3 years on average, depending on your intensity of management change. So if you're going to go gung-ho and you're going to park all the tillage equipment, completely diversify your rotations, integrate livestock, well, then you might run that every other year to evaluate. If you're just going to add a cover crop once every 3 years in your rotation, you might be doing that every 3 to 5 years, right? So the Haney test is then used throughout the year. The idea behind this is that you're trying to evaluate what you have in the soil warehouse, what you have on the low-hanging shelves, what's a low-hanging fruit. And the very next question is always, well, if I've got all this in the warehouse, how do I move it from here to here so I can use it? Much like if somebody called me and said, congratulations, you got $10 million, it's in a vault, it's in an annuity, you can't touch it. Well, what do I got to do to use it, to get it, right? PLFA is generally the one that people start with because it's an introduction. Now, DNA can do the same thing and on steroids. A lot of people start with PLFA just because if they're completely new to this, the DNA reports can be pretty overwhelming. There's a lot there. So again, some people kind of dip their toe with PLFA, start thinking about it differently, looking at things. But yes, in very specific circumstances, you say, well, I'm running plant tissue tests. My plants are always short of calcium. But then you look at the warehouse and you've got this huge amount of calcium and you've got this calcium that's available. Why are the plants not getting it? Well, now DNA becomes more powerful than PLFA.
Sure. Because then you can actually pinpoint down on what actual life needs to get put into that soil in order to release that calcium.
That's right. And a lot of times it's a combination then of, people ask me all the time, do biologicals work? Well, yes and no. Does jump starting a dead car battery work? Absolutely. If the car battery's dead, you see this amazing response. If you hook up the cables right and you do everything correctly, you see this response.
Sure.
But if the battery is already charged, hooking up jumper cables is a waste of time. And so much like if your soil already has these organisms, putting more of them on isn't going to fix that. necessarily. So it's a combination approach between the right biological coupled with the right management. Just like you can go to the gym and exercise all day long, but you can't outrun the fork, right?
That's right.
So you can take all the supplements and go do all the work, but if you don't do this other part, sleep well, try to mitigate, it doesn't help make you healthy. The power is when you combine the two.
100%.
Yeah. And it's the same approach, but we're trying to evaluate those different pieces of the system, and that's where most people start.
What is one thing people get wrong about regenerative agriculture?
That you have to put yourself in a box in order to accomplish regenerative ag.
Perfect. The first step, advice for a conventional grower who is curious but scared to start on the regenerative path.
Start small and start with your worst acres. You have the most to gain and the least to lose. I mean, let that be your playground.
To be able to have your knowledge, your wisdom, what you've accomplished over all of these years. If you don't mind, if you would just let our audience, the listeners, know where they can find Regen Ag Labs, if it's social, whatever that may be.
Yeah, absolutely. Don't try to find us physically because we're in the middle of nowhere. Behind my house. No, I mean, honestly, we're not big on socials. We're not out there much. We do have a LinkedIn page, but I just don't like to manage socials. Best place to reach us is through our website at regenaglab.com. We do rely a lot on just word of mouth and things like this, the podcast and things like that. But I do guarantee you, if you reach out, somebody will get back to you in pretty short order, and we make time to have those conversations. So
absolutely. You guys are willing to change things. And that's why we're so excited as well here at Rocky Mountain BioAg to be working with you guys moving forward. The testing that we're going to get done with you guys and through you guys, and then, the wisdom and the knowledge that we're going to also be able to add to what we're already doing is just, we're excited about it, definitely. But no, I just like to thank you again today, Dr. Gunderson, for coming here, for speaking to our audience. If you are listening to us on Apple Podcasts or Spotify, if you give it a 5-star review, we definitely appreciate it. The Soil Talks podcast, if it's on YouTube, if you would, like it, follow us, take a look. We are bringing the leaders in agriculture, in regenerative agriculture today, to the forefront, and we're showing what these guys are doing. And they are changing the landscape. They're changing everything that's happening. So if you would subscribe to our channel, and we're going to continue to do this. But thank you so much, Dr. Gunderson, for being here, for just sharing your wisdom, your knowledge, and all your expertise. Appreciate your time.
You're very welcome. And thank you, Brandon, and all of Rocky Mountain BioAg for the opportunity. It's been great.
Absolutely.
Really enjoyed the conversation.
Well, thank you. Appreciate it. And you have a great day.
Yeah, you too. Thank you.